Hi, everyone. I'm Jack Jacobson, and I'm an advisor at Thompson Coburn, and I'm one of the Council for Federal Cannabis Regulations advisors. Today's webinar will focus on the effectiveness of CBD and how to best understand real-world evidence and its impact on science and the general consumer. Now I'd like to introduce my co-host for this series, Sarah Chase. Sarah?
Good afternoon, everyone. I'm Sarah Chase, the Executive Director of the Council for Federal Cannabis Regulation, and it's my distinct pleasure to welcome you to the latest installment of CFCR's webinar series. I am overjoyed to introduce you to our three very distinguished guest speakers today, and I encourage all of you here today to ask a lot of questions. You can put those into the audience chat or the audience Q&A, and we're going to try to get to as many of your questions today as we possibly can. So today, we're joined by Pelin Thorogood and Dr. Jeffrey Chen, the two co-founders of Radicle Science, which is a transformative health tech company that offers a scalable path toward predicting and validating the effects of health and wellness products for ailments or the enhancement of human function. And we're also joined by Tim Orr, who's the Senior VP of R&D and the President of Charlotte's Web Labs, a company that produced the now legendary treatment regimen for 6-year-old Charlotte Figge that ultimately helped affect a seismic shift in public opinion toward the use of medical cannabis. So welcome, everyone. Pelin, perhaps you could start us off by introducing yourself and a little bit about Radicle Science.
Sounds great, Sarah. And first of all, thank you again for hosting. It is such a pleasure to be here and talk about this really important topic. So thank you so much to CFCR. So as Sarah indicated, I'm Pelin Thorogood. I'm the co-founder and exec chair of Radicle Science. My background is engineering, data science, big data, and really leveraging evidence-based thinking to change industries, which is what I've done in over my 20 years of career in technology. So what brings me here? And that's about 6 years ago, due to a health crisis in our family that led to the use of opioids for pain management. I was looking for alternatives and, found CBD and cannabis to be immensely helpful. I also realized there was very, very little data on their effectiveness. So that's when I started a nonprofit, a 501c3 called Holistic Research and Education Foundation, a federally approved 501c3. And holistic is with a W because we're looking at whole systems understanding as well as whole plant understanding of how CBD and cannabis might be helpful. We are, I believe, still the largest private funder of cannabis research across the U.S. We work with amazing institutes such as UC San Diego, University of Utah, UC Irvine, and that's actually how I met Dr. Jeff Chen, my co-founder, who was at the time the executive director of the UCLA Cannabis Research Initiative. We've done some great work, but at the same time, research, especially leveraging academia, has been extremely slow and also very, very expensive. And when COVID hit and when everything slowed down to a screeching halt, Jeff and I, looking at our frustrations in terms of getting the kind of data we needed to better understand how these products work for the population at large, we decided to start Radicle Science, which is bringing a virtualized approach as well as a direct-to-consumer approach to being able to deploy rigorous clinical trials on health and wellness products, starting with cannabinoids, to the general public so we can understand how they work and how effective they are for everyday conditions that people are using it for, such as pain, sleep, anxiety, and well-being. These were not necessarily being studied at the academic setting. Typically, we look at epilepsy, which is how it all started, and then autism and Alzheimer's and other very important diseases, but not the everyday conditions that tens of millions of Americans are going to the pharmacy or the drugstore to be able to buy over-the-counter products. Those were not studied. And that's our mission, to really learn from the population at large how these products work, what works for them, what dose works for them, how they're using it so that we can help educate consumers, healthcare providers, and really, society at large as we're a B Corp, a public benefit corporation. So thanks for that, Sarah. And I'll pass it on to, I guess, my co-founder, Jeff.
Yeah, thanks, Pelin. And yes, Jeff, if you could talk a little bit about yourself and your research background, too, with the academic universities.
Sure. Thanks, everybody. Really excited. And thank you, CFCR, for organizing this. Pelin gave a nice little teaser for that. And so my background is, I'm trained as a physician, but I have always been more interested in things that can accelerate the development of new therapies in the clinic. And so after med school, so I actually got my medical degree, my MBA at the same time at UCLA. And right after graduate school, I started looking around and seeing that cannabinoids represented a major therapeutic potential, and that research had been scant because of nearly 100 years of restrictions. And so really wanting to add more data to that whole landscape, I started UCLA's Cannabis Research Initiative, and this is back in 2017. And so we were one of the first universities in the world that actually had a dedicated cannabis research program. And over the next 3 years, we grew that program to eventually dozens of researchers, and across all our projects, there was an 8-figure research budget, and yet lots of complexities to running these studies. Pelin alluded to some of them, but in general, clinical research, very expensive, very slow. And furthermore, as a federally funded institution, we had to follow federal law to the T. So it didn't matter that UCLA is a state public institution in the state of California. It didn't matter that California had legalized medical cannabis in the 1990s. It didn't matter that it legalized adult use cannabis in around 2017. We still had to follow federal law. So we could not study a single one of the cannabis THC products sold in the state of California. We couldn't even allow them onto our campus to test what was in them, looking for contaminants and things like that. Literally, we could not touch it, could not come onto the campus, even though there was multiple dispensaries in Westwood, 1 block away from the university. Even when it came to hemp-derived CBD, we could not study that. And then the Farm Bill passed, and eventually the university said, okay, you can study this, but if you want to put it into human clinical trials, you need to treat it as an investigational new drug. You can't treat it as a supplement, for example. So for all these reasons, talk about the complexity, the cost of clinical research. Now add on the fact that we were only allowed to study a handful of federally compliant or federally recognized sources of cannabis and cannabinoids. Cannabis coming from, at the time, University of Mississippi. Since then, literally, in the last 6 months, the DEA has issued additional licenses. And then when it came to certain cannabinoids, they basically had to be pharmaceutical cannabinoids. And so talk about not being able to study one of the thousands of cannabinoid products that 50 plus million Americans have used or are using. So it was very frustrating in that setting. And so those are some of the frustrations that led to Pelin and I creating Radicle Science. How could we do this research cheaper? How could we do it faster? How could we do it at larger scale? And how can we do it on products that are the products that we need information on, products that consumers are grappling with, that physicians are trying to understand better, that regulators are trying to understand better. All of the trials, just like Pelin, all the things she funded 5-6 years ago, none of it's done. All the clinical trials that we initiated, none of them are done yet. Or some have finished, but the results are still pending. And so, the public is still generally still left in the dark as to the results of these. And so, we are here to push the ball forward across the board. How do we make clinical trials just cheaper and faster in general? Our focus to date has been really accelerating cannabinoid science, because that's where we saw the biggest need across the sphere. If you think about non-prescription health interventions, by far, cannabinoids are in the greatest need right now. Lots of people using them, not a lot of data because of research bans for decades, and still to this day, research restrictions that still make it very, very difficult to do this in official settings like universities.
Thanks, Jeff. That's a good background. We're going to go into a lot of the detail here too about some of the important topics you mentioned. And I want to give Tim Orr a chance to introduce himself too. And Tim, in your introduction, maybe tell us a little bit for the audience members here who are just joining, a little bit about the history of Charlotte's Web too, and sort of how that plays into I think why you're here in the room with both Jeff and Pelin.
Great. Thanks, Sarah, and thanks for having me. So I'm 30 years in the life sciences space, leading technical teams and commercial organizations. My responsibilities span Johnson & Johnson, Kodak Healthcare, and Abbott Laboratories. The vast majority of that was at Johnson & Johnson for about 25 years. It's kind of funny, I think back, there was a time when I was at J&J that the human genome was being discovered. and all kinds of biochemical pathways and new opportunities were resulting from that amazing work. And I thought at the time, well, geez, that's it. I mean, we've kind of learned everything. What else is there going to be to discover? And then fast forward, I moved into the cannabis space. I joined Charlotte's Web about 2 years ago, only to find that the endocannabinoid system is one of your largest regulating systems in your body, and it's largely unknown, undiscovered, not taught in medical school. So we're 2 years into advancing science at Charlotte's Web. There's a huge amount of discovery. As you know, your endogenous endocannabinoid system is crucial for homeostasis and really keeping your internal environment stable and optimal. So there is plenty of opportunity and discovery ahead. In terms of Charlotte's Web history, and I'm glad you brought this up because it really does kind of play into the science and study product. I won't get into too much of the detail. You can Google or go into YouTube about the history of Charlotte's Web and Charlotte Figge. It's really well documented. She truly is a lovely girl, and she changed the world for us in terms of opening our eyes to cannabis wellness. The products that the Charlotte Web products significantly changed and improved her quality of life. In the early days, as Charlotte's Web was getting started, the Stanley brothers were working with Charlotte and other Charlottes. And one of the things about Charlotte is she was very sensitive about slight changes in product. And so it instilled very early on a culture of quality and an investment into the genetics at Charlotte's Web to develop very stable genetics, very sound manufacturing processes, and quality built into every step to ensure the consistency and reliability. Because when that product changed even a tiny little bit, we'd get a call from Charlotte's mother saying, "What'd you do to the product? Something happened." And when I talk about quality of life, I think, we're not a drug company. That's not what we're trying to be. We're not providing medical advice. So when I think about Charlotte and the situation, people tend to want to go to talk about the seizures and what it did to Charlotte's seizures, but there's so many other things that get overlooked, like improvements in behavior, improvements in alertness, improvements in language and communication, motor skills improved, sleep improved, caregiver burden was decreased, healthcare utilization went down. There was improved tolerance of medications, prescribed medications, feelings of stress get better, feelings of discomfort, emotional stability, and the list goes on in terms of improving quality of life with these products. The one thing, and I'll just reinforce that brought me here, is that because of the consistency and the reproducibility of the Charlotte's Web product, it really lends itself to a study product. And that's one of the reasons I was attracted to Charlotte Web is to take these studies further with a really sound product, so that we can continue to learn more and build a foundation not only of safety, but begin to look at effectiveness and how it really works in improving quality of life.
Great. Well, thanks, Tim. That's a great background here for us. A quick reminder for our audience, please, put your questions into the Q&A for us. And now we're just going to jump right into the conversation. So my first question kind of goes out to all of you, but probably specifically Dr. Chen and Pelin to start. Currently the FDA's position on CBD is a fairly gray area, which has left a lot of companies with their hands tied. And to Dr. Chen's point, often pushed down the IND route. So how are you generating safety and effectiveness data at this time?
Jeff, I'll let you start.
Oh, sure. So, yes, you're right that, well, the first thing that I think we would like is some additional clarity from the FDA as to what what exact data would they like to see regarding CBD in order to make a better regulatory decision. We know that CBD, there are clinical studies on CBD. It's gone through an FDA approval process for a particular use case, in this case, Dravet syndrome and Lennox-Gastaut, these pediatric epileptic cases. But what specifically are they looking for? That would help guide us. But on our end, when we came onto the scene, we saw that the biggest gap was getting real-world data on these non-prescription CBD products that, again, tens of millions of Americans were using, and getting data on their safety and effectiveness. So we kind of took our best stab at what we thought was a scientific approach to understanding these products. And so the way that we have run our studies, we always go for large-scale studies. So if you look at, for instance, our ACES study, which I'm sure we'll dive into today, there's 3,000 Americans involved from all 50 states. So we go for large-scale studies to be able to get as confident in the results as possible to eliminate noise and chances of type 1 error. We go for diversity in the patient populace. One of the drawbacks of many clinical trials is it's these homogeneous populations, and America is very diverse, and we know that very diverse folks are using CBD and other cannabinoids. So we go for what we like to call intentional heterogeneity in the study sizes, and we track these studies using validated outcome measures. Some of the areas that we focused on have included quality of life, well-being, pain, sleep, and anxiety in our studies. And so this approach that we take is we're running these studies in the participants' own homes. And so in that sense, it's naturalistic data. It's as real world as you can get. And that's really what we're hoping to do when we came on to the scene. And this is what we've done to date. As far as safety goes, we've been tracking reported side effects and adverse events across the board in all our studies. We're generally finding that reported side effects are generally uncommon, around 10%. And that the most common side effects are usually lethargy or some form of gastrointestinal discomfort, which is actually quite in line with what you see in the clinical studies that have been done on these pharmaceutical grade CBD doses as well. And another thing, a big missing gap in the literature is that the effectiveness of CBD has been established, but for these relatively rare conditions such as pediatric epilepsy, and whereas most Americans are using them for quite common areas, sleep quality, well-being, etc. So we wanted to study these conditions that people are actually using it for, and studying the right products and doses that they're actually using. Again, in these clinical trials of CBD, you're seeing hundreds, sometimes in excess of 1,000 milligrams a day, whereas the average American that's using CBD might be using less than 50 milligrams a day, less than 25 milligrams a day. And those doses haven't been well studied to understand their safety and effectiveness profile. So that's kind of the approach that we've been taking at Radicle to try to add to this literature. And again, being able to turn around studies in less than 6 months from start to finish, as opposed to this multi-year process of how we go about this in the university setting. And it's because of all those friction points and barriers to research that 5 years later, when I started the cannabis program at UCLA, we still don't know much at all about CBD and these other cannabinoids. So that's something that we're hoping to really change.
Excellent.
That's
Pelin,
great.
I understand.
Jump in, Sarah, but go for it.
Okay. Well, I understand that you're also beginning to run sort of more robust clinical trials data at this time too. And maybe you could tell us a little bit about the two studies that we're aware of at UCSD, I think you said, and then also at the University of Utah. Is that correct?
Correct, and that's with holistic. But just to, and I'll absolutely dive into that. But just to, and it's going to be, I think, a perfect way to pass on to that. Like, for instance, last year, very first year in business for Radicle Science, we started and completed 25 randomized controlled trials on cannabinoids, CBD and some of the other rare cannabinoids. We did it over 7,000 Americans, including people in rural areas and very heterogeneous group of people from a demographic and behavior and pre-existing condition perspective, representative of America's population, which is, I think, what we need. And I'm so proud of this because we got here because of what we've seen that is not sufficient in the way we do traditional clinical trials. So both Jeff and I, from very different perspectives, me as the very active venture philanthropist, as I like to call myself, and Jeff as, of course, the physician running UCLA's center, we've been in the traditional clinical trial side, which is, again, doing amazing work. So it's not to put them down at all, but it is very, very slow. So and again, with very small populations. So the UCSD study, which is truly a first of its kind study, I believe it's the largest CBD trials ongoing. We funded $4.7 million to UCSD to study CBD effectiveness for autism. And that trial started in 2018. And it's a full-on a blinded, placebo-controlled, randomized clinical trial with crossover. So every participant is actually acting as their own control as well. And it is not just a clinical trial, but it also is looking at mechanisms of action, trying to understand how CBD works, but actually leveraging some very, very innovative neuroscience and stem cell research as well, in addition to the full-blown clinical trial. So it's great work, and there's nothing like it. Once we find out, we're going to, I'm sure, learn a lot. But it is taking a long time. And the population is 30 boys between the ages of 8 and 12. So again, it's a narrow population. That's how it works in clinical settings, and I think it's going to be great. But what we try to do is that much larger heterogeneous populations at Radicle Science. With a University of Utah study that actually is completed and is actually in the analytics phase, I believe we're going to get the results later this year, so I'm so excited. That is, again, a first of its kind study on personalized brain effects of cannabinoids across the board. And we're working with 40 healthy adults, and the different arms are CBD, THC, CBD and THC together as well, placebo, again, a blinded, randomized crossover study to understand how the brain works and how it lights up and how it functions differently with these different compounds, and if there's specific biomarkers that can be predictive of how people are going to be responsive. So it's going to be an incredible trial. We're collecting lots of safety data, and this information is going to probably light up a lot of research, but it doesn't give information about the everyday condition, as Jeff indicated. We still, tens of millions of Americans are using it for pain, sleep, anxiety, mental clarity, and things like that. And these trials, while great and are going to be very informative at large, they don't provide us the information to decide which products to buy from over-the-counter, because neither of the two trials I mentioned are using over-the-counter products. So that also is a very important part, because population can go buy something, and yet we have not been able to study that, even though we have tried. We've tried through Holistic, and I know Jeff has tried through UCLA, and due to federal regulations, we haven't been able to study that, so we've done what we can by using more pharma-grade products.
Great. Jackie, a couple of questions here. I know we want to talk a little bit about dosing and maybe optimization usage too.
Yeah. Well, actually, first I wanted to follow up on what we just talked about. In the absence of the clinical trials, how is this real-world evidence really helping build consumer confidence in CBD products?
I can take this one.
Sure, go for it.
Sure. So first off, these are clinical trials that we're running. We're just running them in a virtual, decentralized fashion, which has happened to a significant amount of the trial landscape ever since the COVID pandemic. So for instance, the ACES study we were mentioning, 3,000 people were recruited and screened, consented, and then they were randomized to receive one of 13 different types of CBD products, as well as randomized to a control arm. And then we tracked them using validated outcome measures. Many of these outcome measures were actually developed by the National Institutes of Health to track real-world outcomes reported by the participants in a naturalistic setting, like in their own home, right? And in some ways, I would argue that this type of a research approach, using diverse patients, collecting outcome measures that are self-reported by them in a naturalistic setting, you actually might get at more reliable and externally valid and generalizable data than in a very strictly controlled single-site clinical trial, where I just recruit people who live within driving distance and I bring them into my facility and assess them at that moment in time. And how you experience a health issue, how you feel will be very different when I force you to come on my schedule to my facility and I, have someone sit down in front of you that's a research staff and ask you a bunch of questions and fill out, a survey and then send you back on your way. You're going to respond very differently in that moment than when you have these, when you have folks kind of in their own environment. They can kind of anonymously report these things. They're not feeling like they're being judged. They're not feeling, any of that. So that's why we really like to think of this as, the truest kind of real world data that you can get about, and especially because we're using products that are already out there in the market. And so this data is as reliable as you can get to capturing what's actually happening when these tens of millions of consumers are reaching for one of these products, taking it home, using it on their own accord, and experiencing certain health outcomes.
And I cannot overemphasize the point around actual usage patterns. In clinical trials, you are, told to take a specific amount, and that's how the clinical trial works. And that's great because we are trying to control it from all angles. But real world usage, as we all know, doesn't work that way. And that's the real life experience of tens of millions of CBD users. So it was very important for us to tell people to use as directed by the product, but then track exactly how they were using. And not surprisingly, we found out that some people were doubling or tripling doses. And so we know how much they're taking. We're tracking their reported outcomes based on their actual usage, as well as their side effects based on their actual usage. So that's very, very valuable data. That is not something you typically get in a traditional clinical trial setting. So we need both sides. So I think that's a very valuable, aspect of real world data as well.
And one thing I'd like to add is that facts and data matter in terms of building consumer confidence. Consumers are also asking us to help with the education of caregivers, doctors, pharmacists. And this is the kind of data that they need in order to help guide consumers and answer the questions that consumers have. And to your point, the real world data is important. And one of the things that we learned in participating in a recent study that we did with Jeff and Pelin is that less is more. In fact, the lower amounts that were consumed actually produced the better result. And so that's counterintuitive to how most consumers are using the product, because there's a bias to, I didn't feel anything, therefore I should take more, and then keep taking more. Not to mention that the precedent that's been set by Epidiolex, CBD isolate, where the maintenance dose for an average adult on Epidiolex is 1,400 milligrams a day. So people look at that and they say, okay, well, maybe I need to take that amount. And when you take a full spectrum hemp extract, you're taking 40 times as much as you need in order to produce the desired effect. So I think the data, the real world data, is really important to help inform and educate.
We've got a good audience question here that I'm going to ask of you, Tim, first. It's a little bit related to what Jeff was talking about earlier here. And I'll ask each of you to comment on this too. When you're talking about pharmaceutical grade cannabinoids, can you be specific? Since pharma requires an API status, which would mean the use of the isolates versus the whole plant, should growers or the industry be moving forward with the use of isolates for better predictability?
So there's probably a lot to that question. So first off, I think we've demonstrated that with good stable genetics and GMP manufacturing processes and good quality control, that you can maintain a high level of consistency and reproducibility. So I don't think that you need to necessarily limit yourself to isolates and isolates only. there is also a pathway for drug development. It's a botanical pathway. So again, you don't have to just do isolates to pursue a drug pathway if that's what you want to do.
Yeah, Tim, you bring up a good point. Even the FDA, by creating the botanical drug pathway, has recognized that there is something that's missing if you just boil everything down to an API. And so the botanical drug pathway is less than a decade old. It's still relatively new. Only, not that many drugs have tried to go through that pathway. But what's nice about that pathway is you don't have to actually, you're allowed to have many different ingredients or active compounds in the botanical extract. You don't even need to know which one's necessarily responsible for the effectiveness that's being seen. You just have to demonstrate that this thing is effective and is safe. And so I would say that, in the past, moving towards isolates has been the easy, not the easy way, but it's been the tried and true way of bringing a drug to market, isolating the effects, removing some of these other predictors. But I hope that we will start to do more and more research trying to understand. In cannabis, you call it the entourage effect. But if you look at Eastern medicine, it's basically called herbal synergy. And perhaps it's the explanation for why eating whole foods is very different than just drinking powders all day. The powders might have all the fat you need for the day, all the protein you need from the day, but I don't think anyone here is advocating that you just boil down your nutrition to a bunch of powders of like fat, protein, and carbs. You're not going to thrive. Something else is there. So there's things that we can't even quantify. They're just peaks on the mass spec. We have no idea. No one's actually really characterized those elements. And going back to one thing on the dosage front that Tim had mentioned, what's really interesting is there are actually studies both on specifically on CBD that have shown this kind of U-shaped dose response curve where certain higher doses, you actually get less effectiveness in the product. And this has been done in animal studies for, say, things like inflammation, and in human studies for things like anxiety, social anxiety from public speaking. Where you give higher doses, the effect actually wears off. You have less effect at the higher doses. So clearly, there's something else that we still need to dig into here.
Great. Following up on the dosing issue that we've sort of nibbled around a bit today, how are you and your studies determining dosage and usage, and then including an audience question, what about instances of misreporting for at-home dosing? I think, Pelin, you said that folks are being fairly transparent when they're taking more than originally recommended, but could we dig in on that a little bit?
Sure. So one of the things we try to do is really incentivize the participants, which, by the way, are many. So for Radicle ACES, the study that we're primarily talking about, we had nearly 3,000 Americans in that study across multiple arms. So that's a lot of people. And we incentivize them by not just dollars and cents, which is a typical clinical trial outcome, but by giving them their data back analyzed to show them how did this product actually help you across these indices we're tracking for your sleep, for your pain, for your anxiety, for your well-being, for the conditions you endorse that enable you to participate in the study. So they're incentivized to tell us the truth because the data is something they're going to be able to use personally, as well as the reports we provide them. It's something they can take to their doctor and say, I've been taking the CBD and this is how it helped me sleep. So this is very valuable information. So I would say they have no incentive to actually not tell us the truth. And also because we do it virtually, by literally sending them text messages to their phones to answer a few questions every day, we make it super easy for them to be able to complete it versus, driving to a clinic, and maybe having a journal that maybe you didn't keep up and then filling out those notes every day. So by partially making it digitized, it makes it much simpler and also much more auditable and time-stamped and things like that. And also the fact that we tell them the data is going to help your health gives them the incentive to tell us the truth on a daily basis.
Just to add to what Pelin said, right? Yeah, well, it's if you look at traditional trials, especially ones that are site-based, oftentimes you're telling people to keep a medication journal. It's literally you're asking them to write stuff down, and then once every few weeks when they come in, they bring that and hand it, and that's basically the record of their usage. But you have no idea if they're sitting in the parking lot filling out the last 27 days of consumption logs because they don't want to be judged when they walk into the clinical trial site and hand over this journal where there's a lot of blank entries. Whereas when you're collecting things in real time from people directly, like Pelin said, they're time-stamped. We know who filled it out. We can prove it. We can prove it came from their device. And yes, anyone could misrepresent things in a phase 3 pharma trial that's site-based, in a virtual trial like those that Radicle is running. But at least in our studies, we unblind them at the end. We give them their data back in an analyzed report. Whereas in most studies, you're not unblinded. You don't get your data back. So, you add on, in addition to all the friction points of being a typical study, you have much more incentive to actually not be compliant. And the last thing I'll say is, in our studies, you can analyze the data 2 different ways, right? You can look at the intention to treat analysis, which is basically just saying, what was the serving size suggestion on the label product? And it's an intention to treat analysis, because ultimately that's what you can all, even if they end up taking 3 times as much of a dose, the label instructions are all you can really change, whether you're a pharma product or a dietary supplement. So we'll do an intention to treat analysis, and then we'll also look at the actual reported dosages as well to see what additional things that it can inform us. But again, we from the bulk of our analyses, we usually go off an intention to treat analysis, which is pretty standard in clinical trial study design and reporting.
Yeah, and if I could just add, so dosing is linked to intended use, as Jeff was just mentioning, and it starts with dosing for studies for toxicity and safety first. Normally you do that before you put it in human. And then it's dosing for general wellness and dosing for efficacy, which is very specific condition-based. The latter two of which Jeff and Pelin are doing and contributing a body of data to this field. From a toxicity and safe use standpoint, really every manufacturer in this space needs to go through the requisite toxicology studies to ensure that they're using and recommending dosing levels that are well below the toxicity levels and ensure safe use. And that is normally done also with a panel of experts that would review that data to kind of stamp it as generally recognized as safe. And that should be done before any human studies done, and I'd recommend every manufacturer go and do that.
And I've got a kind of follow-up question on that one, because we're talking about toxicity here. What have we learned from some of the toxicology reports that are out there? And Pelin and Jeff, maybe you could talk a little bit about the results of ACEs on the side effects, what's notable in the data. And then to build on that, there's an audience question too about are these studies determining which conditions would benefit from CBD and which conditions wouldn't benefit? Maybe you could talk a little bit about both the results, what's notable in the data, what we found from the toxicology, and then what conditions might benefit from what we found with CBD.
Jeff, you start. I'll add a few things, I'm sure.
Or maybe, Tim, do you want to start with what some of the preclinical tox stuff shows, or whatever the safety data of record shows?
Yeah, so most of the safety concerns really center around CBD, and I mean CBD, the isolate, CBD, the molecule. And that's very different than CBD that's found in a full spectrum hemp extract. Unfortunately, because of the regulations and the way that they are set up today, there are more studies that have been done on CBD, the isolate, than there has been done on full spectrum. So in all of our toxicology work on full spectrum, there was no evidence of toxicity with any of the work that we did based on our intended suggested uses. But if you look at the studies that are done with Epidiolex, and this is where it has become problematic for really people who are following this and concerned about safety, is if you go back to the dosing that I mentioned, the average maintenance dose for Epidiolex for the average size adult is 1,400 milligrams a day. If you contrast that to the average dosing that somebody's taking on Charlotte's Web product is about 35 milligrams. So the dosing on Epidiolex is about 40 times higher than it is for a full spectrum hemp extract. Now, if you have high levels of anything, it's going to become toxic. And what we found with the Epidiolex is that, and this is, you don't have to dig too far in the literature. If you go to the Christopher Pauly study, who looked at a meta-analysis of all the Epidiolex studies that were done, he found that in one study for Lennox-Gastaut, 86% AEs, adverse events. For Dravet syndrome, 93% adverse events. For Parkinson's, 100% for adverse events, and so forth and so on. So the point is that these studies are looking at CBD at pharmaceutical levels. What we're talking about is something that is 40 times lower than that. And so they're very safe, they're very well tolerated. And I think some of the work that's been done with the Epidiolex studies, while great from a pharmaceutical standpoint, has raised safety concerns for these as supplements. But I think we need to see more studies on hemp extracts at the supplement suggested serving size level to really understand it.
Yeah, and that's precisely like what Tim said is right on the nose. And as far as, again, in our studies, we are collecting adverse event information. In this particular study with ACEs, we weren't tracking liver function tests, but other studies have looked at liver function tests in people taking these real-world doses of real-world commercially available CBD products. And there didn't seem to be any liver function elevation of liver enzymes, aka no suspected liver damage at those doses using those products. In our studies, we were mainly tracking self-reported adverse events and side effects. And again, across the 3,000 people in ACEs, for example, about 10% reported any side effect due to the product, and the most common one were some sort of gastrointestinal discomfort, maybe some diarrhea, some upset stomach. And the other ones were reporting some somnolence, so they were a little tired from the product, which is, again, not unexpected. And 10% as a side effect rate is, especially with these mild side effects, is not, at least from that standpoint, it's not something that's particularly alarming. Now, effectiveness-wise, if you look at the prior clinical trial data, we talked about pediatric epilepsy. We talked about pediatric being one, but some of the other areas where CBD has demonstrated effectiveness, it's everything from showing it has like anti-addictive properties in certain instances. These are human clinical trials run in academia. So, anti-addictive properties, helping schizophrenics with their psychosis, helping certain forms of anxiety, in particular social anxiety, little bit of data on it modulating sleep effects. But what we really hope to find, and what we found with ACEs, we hope to find with subsequent studies, is really digging into the main reasons that your average American is using cannabis, using CBD. And if you look at the polls of CBD consumers, you find that the top three generally are to help improve their sleep. Maybe they're having discomfort in some part of their body, or maybe they're stressed or having feelings of anxiousness. And so those are the areas that we really wanted to focus and hone in on in ACEs. And what we saw in ACEs was there were significant improvements across the board in those areas, and specifically clinically meaningful improvements. So in both for pain, sleep, and anxiety, we saw, at least close to 50%, some instances well past 60% of participants seeing clinically meaningful improvements in those areas. And so these doses, these products, these common conditions that people want to use it for, these need to be studied if we're going to have a coherent conversation about how best to have these products regulated, how best to sell these products.
Thanks, Jeff, for that. And one of the things I wanted to add is we talked about clinically meaningful results, statistically significant results that are focused on the endpoints we were studying, understanding CBD's effect on well-being, sleep, anxiety, pain, etc. And that's great. And that's, again, using these validatedness is a wonderful way to start. But because we have this massive data set, 3,000 people versus your traditional, maybe dozens, maybe 100 people, we also have the opportunity to use machine learning, a form of artificial intelligence, to be able to dive into the data, to look for hidden correlations, to understand, are there differences between people? Are demographic differences or behavioral differences making providing a variation on the theme of how people are seeing the effects, or do they need different doses? Are they seeing different types of effects? So our goal with Radicle Science, in addition to studying the effects on average, how these products may affect the public, which is very important from an epidemiological or public health perspective, we're also trying to get to precision medicine and really understand how different people based on demographics and behaviors may actually need different doses or different types of products. So that's another area we're going into. And the size of our data set, the heterogeneity of our data set actually enables us to go there, which is very exciting.
Great. Thank you, Pelin. We've got a couple minutes, about 15 minutes left. So if you have questions, please submit them and we'll try and get to them before this distinguished panel is through. Dr. Chen, could you talk to us a little bit about what bioavailability is and why it's important in this context?
Sure. So bioavailability is described as the amount of a ingredient, a drug, a substance, a product that actually makes it into your bloodstream. And once it's in your bloodstream, it can actually circulate in your body, it can be absorbed by cells, it can reach different organ systems. And so generally, what we start with percentages, and it's a benchmark. So 100% bioavailability, you achieve generally through IV injection. So if I IV inject something straight into my blood, it's 100% bioavailable. Every bit of that thing that enters my body is sitting in my blood and ready to be uptaken by my cells and will make it to my organ systems. Other ways that things can reach our bloodstream, you can inhale and smoke something. Bioavailability is generally high because your lungs and your bloodstream are very interconnected, and there's no, say, liver in between, and there's no digestive process and absorption process. Absorption via oral intake. So when you swallow a pill, generally you get lower bioavailability that way because you first has to enter your stomach, go to your small intestine, be broken down, absorbed, and then still has to make it through the liver. And sometimes the liver will break down a certain percentage of what you just swallowed, and then only then can it get dumped into the bloodstream. So when it comes to all cannabinoids, bioavailability is generally poor when it's just on its own. So if I just took pure CBD and its crystalline powder form and swallowed it, I'm not going to absorb very much at all. Like low single-digit percentages will actually make it into my bloodstream. There are ways to increase bioavailability of things like cannabinoids, in particular CBD. Oils and fats will increase the bioavailability. So it can be whether it's dissolved in oil in a tincture or whether you take cannabinoids with a meal that's been shown to boost bioavailability. But the reason this is so important is because it means that not everything's created equal. You could have the 25 milligram CBD products, and you can have 10 of them, and depending on their formulation, you could have dramatic differences in how much of that 25 milligrams actually ends up in your bloodstream.
We have a couple of questions that are coming in from the audience here that are applicable to this too. So one of the first ones is, have any of the studies that you've done used emulsified cannabinoids or terpenes?
Terpenes, yes. The studies we've studied, full spectrum products where we've studied both full and broad spectrum products where there are terpenes present. When we say emulsified, that probably needs a additional clarification because there's many ways to emulsify products. But generally, yes, we have studied products that have used emulsion systems in the form factor as well.
And then for clarity here, and this is probably for you, Dr. Chen or Pelin, what average daily dose of CBD worked best for anxiety, sleep, and pain that you found during the studies?
So one thing that we have, we've released some preliminary data in aggregate across our studies. We have also described in general the doses that people are using, but what we've been hesitant to try to associate actual usage with the outcomes observed. And this is not just unique to us. This is across the sphere in other clinical trials as well. And it's because you start hitting this notion of reverse causality. Is someone increasing their dosage because they're just by the pure natural course of their condition, it's the symptoms are getting more severe. And that's why maybe they're having a really bad week and their sleep is especially bad. And that's why they're increasing their dosage. Or are they increasing their dosage, seeing a beneficial effect and increasing the dosage more? And so because you can't disentangle that, it can be a little misleading and hard to interpret actual consumption data with the outcomes, which is why you often go off intention to treat analysis. What was the suggested serving size on the label of the product? What were the health outcomes actually seen? Because ultimately, all you can really control at the end of the day is the suggested serving size that sits on the product.
Pelin, Tim, anything to add to that?
I think Jeff covered it.
Yeah, I think you got it.
Maybe you could briefly, before we get to the sort of last end question here, maybe you could talk a little bit about just some of the key highlights from the data, the key takeaways that we should know about.
Sure. Now, keep in mind, the first thing, I'm not sure who to attribute this quote to, but they say every great study warrants like 4 follow-up studies. So this was the first of many studies to come. We only studied 13 products out of a universe of literally probably over 1,000 different CBD products out there, if not more, thousands, plural. But some of the key highlights that we saw is, again, significant and meaningful improvements across the board, pain, sleep, anxiety, using these validated outcome measures. If you had to rank them, it was anxiety saw the greatest percentage of people having clinically meaningful improvements. Next up was sleep, and then after that was pain. You saw improvements across the board for well-being as well, overall well-being. We found, and Tim alluded to this earlier on, what we found was that in some instances, the lower dose products actually were outperforming the higher dose products. And that partially could be explained by what we were talking about, say, maybe bioavailability or the formulations, right? Just because 2 products both have 25 milligrams doesn't mean you'll absorb the same. That also doesn't mean that a 25 milligram product will always beat a 10 milligram product. Depending on how they're formulated, this 10 milligram product might, more of it might be reaching your bloodstream than the 25 milligram product. So that was an interesting finding as well. When we analyzed the products by spectrum, full, broad, and isolate, we had kind of all 3 types of products represented, we didn't see a significant difference when separating by spectrum. But again, this is 13 products out of a universe of, thousands. So I think it's still up for debate as to how the different spectrum products, how the entourage effect actually works, and in which products it's actually at work. But those are some of the kind of major kind of findings from the study.
I have a couple to add, and I really, these 2 are very meaningful for me. First of all, we saw improvement across the 4-week study, but the largest improvement actually happened within the first week. So there was a significant improvement across all the indices Jeff mentioned within the first week. And then people continue to improve throughout the 4-week study. So that was really good to see. And the other one is the onset of the effects. And we saw very quick onset. People had to respond indicate, when they started feeling the effects in their patient-reported outcome data. And across the board, within 1 to 4 hours of taking whichever product across the board, the participants were actually seeing an impact across all the areas that we're studying. So it made a difference within taking it, and they saw very significant improvements within the first week with continued improvement throughout the 4-week study.
And just to add some quantification to what Pelin and Jeff has said, in again, my lens, I'll look through the Charlotte's Web products, not necessarily the entire study product, but for Charlotte's Web, there was, of the 189 participants with Charlotte's Web, there was a 74% increase in well-being. And so it's a good power score, the 189, so it's statistically significant. And to have 74% of the participants with an increase in well-being, I mean, that's a meaningful shift. 32% improvement in sleep quality, 53% decrease in feelings of stress, and a 28% decrease in discomfort. So just to add some quantification to what you've said.
Thanks for that, Tim.
That's really helpful, Tim. And I'm gonna ask the last question, but all of our panelists, I encourage you to offer closing remarks here. But what did we learn, and what do we need to learn more about in order to really ensure health and safety around these products moving forward? Pelin, you want to start?
First of all, in terms of what I would really like to, we're doing a lot. We're doing what we can do, both on the traditional clinical trial side, with all the organizations we're working with, including the studies we're funding through my foundation, Holistic Research, as well as obviously doing all these real-world studies with full rigor at Radicle Science. Would really like to better understand exactly what FDA is looking for, especially with these over-the-counter supplements that people are actually able to buy. Tens of millions of Americans are buying it. We would like to understand what additional data they're looking for, both on the safety side as well as the effectiveness side, so that we can put our energy and dollars and time in doing what the FDA is looking for to be able to get the data so they can make the right decisions. To me, this is really important, especially given the massive population that is using these products today, and we want to make sure that we're providing the information to the regulators and also have the information for just public health in general.
Yeah, and if I could add, I'd like to see studies move beyond healthy populations. So when you're studying general health and wellness, it's generally studies on healthy populations. We have consumers that have conditions, and they'd like to have condition-specific data that they can go to. And that also plays into the education need for doctors, caregivers, pharmacists who also want that information because they're being asked by consumers. So I'd love to see, when you get into a diseased populations, that's where it gets a little bit difficult with the regulations and being able to do the studies without having to file an investigational new drug. So it's been a tough regulatory environment from that standpoint to do more of the studies that I think the industry would like to see done.
Absolutely, Tim, and really doing studies on the formulations people buy and use every day instead of these, molecules, which I know we need to study, and this is not instead of, I'm talking in addition to, so that we can actually look at how it's being used so we learn from that, in addition to doing the far deeper dive with academic research. I think both of these have to be concurrent because we learn very different things, and both types of studies can inform each other to accelerate the overall data and insight that we need as consumers, as healthcare providers, and of course, as regulators.
Yeah, I think Pelin and Tim nailed it pretty hard. I just want to point out that, just a reiteration of what they said, it seems a little ridiculous that when you're trying to do research where you're monitoring someone's safety, you're sending them product that you've tested and you know what's in there for the purpose of gathering knowledge for society. You just want to do research on these products, that somehow you're forced to go down an investigational new drug pathway, which immediately adds an order of magnitude to the cost and time to completion to gather knowledge. Yet, tens of millions of Americans can walk into stores or buy these products online, and there's thousands of products out there. So that it seems it's pretty frustrating to see that quandary, and it's not serving anyone. It's not serving the regulators who need more knowledge and data. It's not serving the researchers who want to understand these products. It's not serving the consumers who want more information about what conditions may or may not be impacted by these products. It's not serving the healthcare providers. So I just hope that, at least in the spirit of having more knowledge and allowing more research, that there should be this IND requirement just to do studies on CBD shouldn't be in place. And that's the only way we're going to get accurate information out there, so that businesses, consumers, doctors, and regulators can make the best health and business decisions as to what to do with this industry.
Excellent.
Yep, we need more clarity on what to study, and we need a pathway to study all these products that are available out there that people are actually buying, and being able to study them for safety and efficacy, just so that we can actually do the public good that's needed.
Excellent closing. For those who are still with us, we have a short survey at the end. We'd appreciate if you take it really quickly as you close out. And our next webinar will be Thursday, April 28th at 1 p.m. Eastern. We'll be talking about medical use cannabis and the safe and legal path forward. And we're excited to be joined by representatives of the Epilepsy Foundation, Dr. Peter Grinspoon, and representatives from both Senate Majority Leader Schumer's office and Representative David Joyce's offices to talk about federal legalization and the work of patient advocacy groups for advancing medical use cannabis. Sarah?
And I can't thank you all to our panelists for joining us today. This was just a wonderful and insightful conversation. So thank you again for being here. As Jack mentioned, our next webinar is going to be very timely with representatives from Schumer and Joyce's office joining us. So save the date, Thursday, April 28th, and you can go to www.uscfcr.org/events for more information. Pre-registration will open in about a week. And also a video copy of today's webinar will be posted on our website, so you can also share it with anybody who wasn't able to attend. And that website again is uscfcr.org/events. Thanks, everyone.
Thank you so much, Sarah and Jack. Really appreciate it.
Thanks for having us.
Take care.
Bye.